Welcome, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia series and our sponsor, Quanterix, I'd like to welcome you to novel CNS biomarker applications enabled by Simoa technology, tau in preclinical and clinical studies. I'd like to introduce our moderator for today. He is Dr. Andreas Jeromin, PhD, CMO, Consulting for Quanterix Corp. Welcome, Andreas.
Thank you, Elizabeth, and again, welcome all to this webinar on preclinical applications of tau and clinical applications as well. We have secured two outstanding speakers today in the order of presentation. Stephan Kaeser from the Hertie Institute in Tübingen in Germany, who will start out on the preclinical applications, the development of an assay for marine tau, and then followed by Mielke, who is with Mayo Clinic in Rochester, Minnesota. So without further ado, Stephan, if you would take literally the ball and start the presentation.
Thank you, Andreas. It's a pleasure for me to present some of our recent data on CSF tau in mice. The title of my presentation is Assay Development for the Quantification of Murine Tau. Before I start, let me give you a brief overview on microtubule-associated protein tau. Tau is mainly expressed in neurons of the central nervous system. In the human adult brain, we can find 6 different isoforms, as you can see here, as a result of alternative splicing of exon 2, 3, and 10. As the name already suggests, tau binds to microtubules and is therefore an integral part of the cytoskeleton. However, it has also been shown that tau can be released into the extracellular space even under physiological conditions, and therefore can be found in the interstitial and cerebrospinal fluid. There are several studies indicating that the most abundant tau species in the extracellular space are CSF, and the CSF are the N-terminal fragments and not necessarily the full-length or C-terminal fragments. Hyperphosphorylation of tau leads to one of the hallmark lesions of Alzheimer's disease, the so-called neurofibrillary tangles. Moreover, Alzheimer patients show increased CSF tau levels compared to healthy controls. In fact, tau is, besides A-beta 42, the most thoroughly investigated and validated CSF biomarker for Alzheimer's disease. So, in cross-sectional data from patients with familial Alzheimer's disease, CSF A-beta 42 with a certain delay, also tau changes can be observed decades before the actual onset of clinical symptoms. So this renders them promising biomarkers for early phase of the disease where therapeutic intervention is considered to be most effective. A-beta 42 decrease in the CSF is probably related to the aggregation and deposition of A-beta in the brain, the so-called sequestration hypothesis, whereas CSF tau increase is thought to be a consequence of extracellular tau release upon formation of neurofibrillary tangles and also neuron loss or degeneration of neurons. However, it turns out to be difficult to correlate these brain lesions to CSF biomarker changes observed in patients based on CSF measurements during lifetime versus neuropathological assessment post-mortem. Therefore, we actually started more than 5 years ago to collect CSF from APP transgenic mice and measure A-beta and tau in order to correlate it directly to the cerebral beta amyloidosis that is seen in those mice. So let me quickly introduce the mouse models we mainly use in our studies. This is actually the APP-PS1 mouse model that was generated in-house, which overexpresses human APP with a Swedish double mutation and human mutated presenilin 1 under the control of a Taiwan promoter cassette. On the other hand, we used the mouse model, one of the mouse models from Novartis, the APP23 model, which overexpresses human APP with a Swedish double mutation. And both of these mouse models develop plaques with aging, but it's important to mention here, they do not have neurofibrillary tangles, and they also do not show overt neuron loss. So what we saw basically around about 3 years ago is that when we do the measurements of these CSF biomarkers in these two mouse models, they nicely reflect what we see in the human. So they also show a decrease in A-beta 42 in the CSF when they basically start to develop plaques, which is in the APP-PS1 model at about 1.5 months of age, whereas in the APP23 it's between 6 and 8 months of age. With a certain delay, at least in the second mouse model, we also see a CSF tau increase in these mice. The orange and green curve here just should represent the brain A-beta deposition. So that's what we have published 3 years ago, and at that time we used for the tau measurements a commercial assay from Mesoscale Discovery, which was the only one that we were aware of that was able to measure murine, endogenous murine tau, because luckily the antibodies that they use in the assay would also recognize the mouse form. However, one of the big problems that we had at that time basically was that the most levels of the non-transgenic mice and the ones from the young transgenic mice were below detection limit, so which was around 35 picogram per ml. And since we have to dilute our CSF samples from the mice like 14 times, this accounts for about 500 picogram per ml detection limit. And as you will see in a few seconds, this is actually the levels are below these 500 picogram per ml. So what we then did when we heard for the first time from Quanterix and their high sensitivity platform, the Simoa array, we got a lot of antibodies from partners from the academic field, but also from the industry, and tried to set up a sandwich assay. We basically had 5 N-terminal anti-tau antibodies, 7 that will bind in the mid-domain, and also for a control C-terminal antibodies, although we basically assume that the C-terminal ones, since we have predominantly N-terminal, would not work for CSF tau measurements. We always assessed these antibodies on Western blot, so we used brain homogeneous from tau transgenic, tau knockout, and wild-type mouse in order to see whether the antibodies cross-react, because most of these antibodies are generated against human tau. So an antibody that would cross-react would typically look like that we have in a tau transgenic, but also in a wild-type mouse, you would see a band or several bands that represent the tau, whereas a human-specific one does not show a band in the wild-type brain homogeneous. Hopefully, in the knockout, both of the antibodies will not show any result. So the antibody pair selection then was done on a Luminex platform, just because at that time it was already feasible to do multiplexing. And with the 14 antibodies that we use, basically we would come up with almost 200 possible combinations and orientations that we tested. And at that time we were just looking for the most sensitive one, since we were not sure whether we are really sensitive enough to detect the CSF levels CSF tau levels in our mice, especially in the non-transgenic and young transgenic ones. So with the most promising one then were transferred on the Simoa platform, and it turned out that ADX202 from ADX Discovery seemed to be the best capture antibody in our hands, in combination with a 7079 from BioLegend, which turned out to be the best detection antibody. And both of these antibodies, as you can see here, bind in the proline-rich domain. And a typical standard curve after proving the assay looks like that. Down here we have we calculated an LLD of 340 femtogram per ml and an LLOQ of 690 femtogram per ml, which turned out to be totally fine for our purposes. So then we went on, did assay validation, of course, intra-assay variability was tested and gave a nice CV. Also, inter-assay variability seemed to be fine for the whole range where we use it. Spike recovery, although we did not reach the plus minus 20% when using peak plasma, which turned out to produce the least signal as a matrix in our assay. So we used this and spiked it with recombinant murine tau, and we always got the same recovery, which was fine with us. So we went on, did parallelism assessment, of course, because we, due to the low amounts of CSF, but also blood that we have basically to dilute our samples. Also, you would like to do that because you would expect matrix effect otherwise. Therefore, we tested parallelism, and it turned out that for CSF tau we can dilute up to 80 times what we in fact do for our CSF samples, because we normally do 5 microliter aliquots. Therefore, we have to dilute them 80 times in order to measure duplicates all the time. And for the blood plasma, we got higher volume, so we can work with 8 times dilution, which is totally fine. We could even go lower. So then we tested our first big cohort of aging cohort of APPPS1 mice in different age groups, starting from 1.5 months up to 19 months of age. And we clearly see that in the transgenic mice, which are represented by the solid dots, that you have a nice significant increase with aging, whereas for the non-transgenic ones, which are magnified in the upper right panel here, you see first a little decrease and then a subtle increase up to the level that you see in the very young mice. So it is, if at all, a neglectable increase in those mice around these 200 picogram per ml. What we then, of course, did as well, we measured brain A beta in those mice, and we wanted to see how the correlation is. As you can see here, it's a very nice correlation, and you also it depicts a linear regression, basically, when you look at the profiles from depicted in the green curve, the brain A beta, and the blue curve represents the CSF tau. You see with aging, the percentage increase is really looking pretty similar in those, which lead us to the hypothesis that CSF tau in this mouse model nicely represents the beta amyloid increase in the brain. So we then, to further challenge our hypothesis, we then went on and used the potent BACE inhibitor from Novartis, the NB360, which has been shown before to prevent the production of A beta, so to decrease a beta deposition and aggregation, and also to decrease soluble A beta levels in the CSF. And we took our mice, the APPPS1, started the treatment when they normally start to develop plaques at 1.5 months of age for half a year, with food pellets, one group with the BACE inhibitor and one group with the control pellets. And as expected, we could see that the increase of A beta deposition in the brain is basically prevented almost completely, which is also reflected in the measurements of A beta, biochemical assessment of brain A beta. On the other hand, CSF A beta 42 is already decreased in the control group, which actually reflects the sequestration of the A beta 42 to the plaques, and therefore the lower levels of of the A-beta 42 in the CSF, and therefore BACE inhibitor in that mouse model was not able to further decrease CSF A-beta 42 levels. On the other hand, CSF tau shows a clear picture where we see that there is a complete prevention of the CSF tau increase that we would normally expect in the control mice, in the APPPS mice at 7.5 months of age. The same more or less was seen in the APP23 mouse. This model we only started to treat with 15 months of age. So in that regard, we had a model that already, as you can appreciate here, had moderate plaque deposition. And as expected, we could prevent further increase of plaque deposition, assessed here by stereological quantification, but also by biochemical measurements of A-beta. CSF A-beta 42, as I mentioned already, looks different in that mouse model because there is only a subtle decrease of A-beta 42 in that model in the CSF, because the deposition is just not as strong as in the 42. And with BACE inhibition, you could even further decrease it. So there's a completely different, or not completely, but a different picture in compared to the APPPS1 model. Whereas the CSF tau again shows a clear-cut picture where you have a prevention, complete prevention of CSF tau increase when treating with BACE inhibitor. Just to rule out that we have an effect, a direct effect of the BACE inhibitor on CSF tau, we also did a short-term experiment where we basically treated the mice just for 1 week, and again, as expected, CSF A-beta 42 would go down immediately in both mouse models, whereas CSF tau remains more or less the same, indicating there is no such a direct effect. So what clinical relevance do these data have actually? So hopefully we will find out in a few years, because all the big companies that do these clinical trials with BACE inhibitors have as a secondary outcome measurement CSF tau included. So we hopefully will see a same effect in the humans, further proving that the mouse model was really instrumental in predicting such an effect of BACE inhibition. Conclusion, the homebrew tau assay, our new homebrew tau assay, allows the quantification of endogenous tau in virtually any murine sample, CSF, blood, but also brain, of course. CSF tau levels in the APPPS1 mouse model are strongly correlated with the cerebral aggregation of A-beta, providing further evidence that the beta amyloidosis is a main driver for CSF tau increase, and prevention of this age-dependent CSF tau increase in the APP transgenic mice suggests indeed a downstream effects of BACE1 inhibition. And last but not least, a tight correlation between a beta aggregation in the brain and the tau levels in the CSF render CSF tau a valuable marker to predict the effectiveness of BACE inhibitors in current clinical trials. So with that, I would like to thank all our collaborators that were really helpful to come up with such an assay. Thank you.
Thank you, Stephan. So we're holding questions till the end. I already seen some coming into the chat room, and our next speaker now is Michelle from the Mayo Clinic in Rochester. Michelle, please.
So thank you, Jeromin, for giving us the opportunity to present some of our findings. And also thank you, Stephan, for introducing the tau pathology and relationship in Alzheimer's disease. So I'm going to go kind of to the other extreme from what Stephan was talking about and discuss some of the work looking at plasma tau in clinical samples, both as a potential diagnostic or prognostic marker. As Stephan showed, CSF tau is really one of the critical elements in terms of predicting Alzheimer's disease and also potentially being used as a diagnostic marker. And tau actually correlates much better with cognitive symptoms than amyloid does. So from that sense, it would be great from a population perspective to trying to determine who's going to progress, and even for clinical trials among those individuals that are amyloid positive, who's going to progress. But trying to do a lumbar puncture or tau PET imaging on everybody is infeasible. So we've been working using the Simoa technology with Quanterix to try and look at plasma tau and understand the feasibility of that as a biomarker. First, I'm just going to give a very brief overview of some of the plasma tau studies to date for Alzheimer's disease and other neurodegenerative conditions, discuss some of our results, and then also discuss some of the experience we have using the Simoa technology in collaboration with Quanterix on inflammatory markers, which we have found to be much better than some of the MSD platforms. So actually, looking back in the literature, plasma tau was first looked at about 15 years ago, particularly using ELISA techniques. And the early initial studies did not find a difference between dementia and controls for plasma tau levels, or actually found reduced tau levels. But more recently, using the Simoa technology, other studies are reporting higher plasma tau levels in several neurodegenerative diseases. And up on the screen are just a couple of those citations, including after cardiac arrest, acute stroke, and more recently there have been a few papers looking at plasma tau and TBI, suggesting that's elevated after injury. The first real study in Alzheimer's disease by Heinrich Zetterberg and others in Sweden, and they had cross-sectionally looked at plasma tau levels in cognitively normal individuals, those with MCI, as well as Alzheimer's disease. And then longitudinally among the MCI individuals, they looked at plasma tau and compared among those that progressed over about 100 months compared to those that didn't progress to dementia. And again, they also used the Simoa technology for this. So initially, Heinrich and others had found that while there was no significant difference between cognitively normal individuals and MCI, the individuals with Alzheimer's disease did have significantly higher levels to both the MCI and controls. However, when they looked longitudinally, there was really no difference among MCI for those that progressed to Alzheimer's disease to those that remained relatively stable over about the 5 to 6 years. Further, they had also, in this specific paper, looked at the correlation between plasma tau and CSF total tau and did not find a correlation. However, of note, their CSF levels were conducted using the Luminex platform, and therefore there could be some differences just in the results due to the differences in platform. So building off Heinrich's work, we wanted to further explore the utility of plasma tau either as a diagnostic or prognostic marker, and also to understand how it's associated with demographics and other medical conditions just within the general population. And so for this, we included 539 participants that were enrolled in the Mayo Clinic study of aging, including both cognitively normal individuals and individuals who had mild cognitive impairment. All these individuals had concurrent neuropsychological measures as well as amyloid PET imaging, FDG PET imaging, and MR imaging, including hippocampal volume as well as cortical thickness measures. And our quantification of abnormality on the neuroimaging measures have been published in the past, but are based on the 90th percentile of AD subjects from the Mayo Clinic. Jeff Dage at Eli Lilly measured the plasma tau, or conducted the plasma tau assays for us, again on the Quanterix Simoa HD1 platform. We used the mean of 3 replicate measures and had a coefficient of variation of about 10%. Now, when we first looked at the data, it was skewed to the right, so we did log transform the data before doing any of the statistical analyses. When we first looked at the levels, our levels were very similar to those that were published by Heinrich, both for the cognitively normal and for the MCI individuals. So I believe Heinrich's mean levels were about 4.2, and I think ours were about 4.1, so very consistent across studies. So the first thing we did was look at the association between plasma tau and odds of MCI cross-sectionally. And what you can see is that higher levels of plasma tau were associated with increased odds of MCI, but the results were not statistically significant. So in multivariate models, we ended up with a p-value of about 0.18 for continuous measures, and then comparing the highest quartile to the lowest quartile with about a 1.6-fold increase odds, the p-value again was 0.09. One thing I do want to mention about this, as you can see, we looked at several different models, the first unadjusted, the second for general demographics. and the third including BMI medical conditions, specifically cardiovascular conditions and cardiovascular medications, because when we first looked at factors that were affecting plasma tau in the population, we did find that those with cardiovascular disease did appear to have higher plasma tau measures. And so that's something that we're further pursuing in other research. So again, there was a suggestion of higher plasma tau in MCI, but not statistically significant. We next looked at the association between plasma tau and domain and global Z-scores. And here we did find that, again, higher plasma tau was cross-sectionally associated with better performance in global cognition as well as better performance in memory Z-scores. Now, adjusting for all covariates in global cognition, it wasn't statistically significant, but among the cognitively normal, but in Model 3 for memory, higher plasma tau was associated with lower memory Z-scores. And this association appeared to be specific for memory, as we did not find significant associations in other cognitive domains. Lastly, we looked at the cross-sectional association between tau and our imaging measures. We did not find cross-sectional associations between amyloid PET or FDG PET measures. But among our MRI imaging measures, including both hippocampal volume and cortical thickness, higher levels of plasma tau were associated with abnormal cortical thickness, which means greater cortical thinning, even after adjusting for variables in our fully adjusted model. So finally, we wanted to further pursue and determine whether plasma tau was more specific cross-sectionally to individuals that had amyloid pathology. And so we stratified individuals both by their cognitive status, so cognitively normal and MCI, as well as whether they had significant elevated brain amyloid, so amyloid positive, or whether they had significant neurodegeneration, defined either using cortical thickness or if they were abnormal on an FDG PET 80 signature region. And what you can see pretty much across the board is that there were really no significant differences between individuals with amyloid pathology compared to those without, and by cognitive status here, based on the biomarker stratification, which indicates that this is not necessarily going to be useful as a diagnostic biomarker cross-sectionally. So in summary of these results, I think that there is suggestion that of a cross-sectional trend for plasma tau and higher odds of MCI. And again, it is nice to have consistent levels with Zetterberg's study. We also did find a significant association with the AD signature cortical thickness as well as with memory, so suggesting plasma tau may be associated or indicative of something going on with the AD process, even though we did not find that it would be a useful clinical cross-sectional biomarker. So we are currently working on looking at longitudinal analysis, which I'll present in a second. So even if it's not a diagnostic marker, can we use this as a prognostic risk factor or biomarker among individuals that are amyloid positive? We also need to look at plasma tau longitudinally to see whether change in plasma tau is associated with change in cognition. And then thirdly, and we've just received this data and are analyzing, we're also looking at the association between plasma tau and tau PET imaging. So as I mentioned, Heinrich, Zetterberg, and others had not found an association or correlation between plasma tau and CSF tau. However, those were, it'll be interesting to look at the relationship between plasma tau and tau PET. So as I mentioned, longitudinally, we're just starting to look at among these same individuals, and of the initial individuals that I presented cross-sectionally, we had 478 that had at least one follow-up visit, and on average, approximately 3 years of follow-up. And so we conducted Cox proportional hazards models to look among individuals that were cognitively normal whether plasma tau predicted risk of MCI, and then among individuals that had MCI, whether it predicted risk of developing dementia, including both Alzheimer's and all-cause. We also wanted to assess whether there was an interaction with elevated amyloid, again, to help determine in clinical trials whether this could be a useful prognostic marker, but to also just assess the interaction between the plasma tau as a potential indicator of Alzheimer's pathology. So this is a Kaplan-Meier plot among cognitively normal individuals and risk of MCI. And as you can see, this the solid line is an indicator of the lowest tertile, And individuals that were in the middle and highest tertile of plasma tau had an increased risk of developing MCI. And in multivariate models, if you were in the second tertile, you had almost a 2.5-fold increased risk of developing MCI over the follow-up. And if you're in the third model, you had about a 2-fold increased risk, which again were significant in multivariate models. However, in other analysis, we did not find an association between plasma tau and risk of dementia, both all-cause or Alzheimer's disease, among individuals that were already MCI. We also did not find an association between elevated brain amyloid and tau in predicting cognitive decline either. So again, it appears that higher levels of plasma tau are associated with risk of MCI among individuals that are cognitively normal, but not among individuals that, or risk of progression among individuals that have MCI. Our results are, longitudinal results are somewhat in contrast to Matson and others who recently published a paper from ADNI, where they had found that plasma tau was most significantly associated with cognitive decline later in the disease. So among individuals that had MCI, and specifically among those that had Alzheimer's disease, plasma tau was predictive of cognitive decline. However, our results are more similar to the initial plasma tau study, where Heinrich Zetterberg had also not found an association among individuals that were MCI, and essentially plasma tau was not predictive of dementia among those that were MCI, similar to our findings. And then again, we did not find or there was a lack of interaction between plasma tau and brain amyloid, which indicates that plasma tau is not specific to Alzheimer's disease. And this is not completely surprising given the fact that tau pathology is observed in many other dementias, and I think strongly suggests that we need to look at plasma tau in relation to a variety of neurodegenerative conditions. Lastly, I'm just going to switch topics and show you a few highlights of some of the work we're doing with some of the inflammatory markers. And just to give you a little bit of background, I was putting together some pilot data for an R01 submission examining plasma sphingolipids and plasma inflammatory markers. And as pilot data, we had incorporated an MSD 10-plex pro-inflammatory panel, which included 524 EDTA plasma samples. Unfortunately, we had a much lower yield than what we were hoping for. So among the individuals that we had with IL-6, of these 524, only about 80% had usable data. For TNF-alpha, we did have data on most individuals, but IL-10, we actually only had data on about 75% of individuals. The pilot data, though, was helpful, and we ended up getting the grant funded, but we weren't really happy with the MSD work, so we started working with Quanterix to measure IL-6, TNF-alpha, and IL-10 in about 2,400 plasma EDTA samples and 953 CSF samples. And we were much happier with the performance. So looking at the plasma data, 96 to 97% had usable data. And even among the CSF, we had about 96 to 97% usable data as well. We did compare, there was a good subset of a couple hundred people that we had both MSD and Quanterix on, and we looked at the correlation between the two. It wasn't very well correlated. We were also quite impressed with the interclass correlation coefficients among individuals. So typically with inflammation, with the MSD platform and a couple other platforms I've seen, the variables tend to jump around quite a bit when you're looking at serial samples. And using this specific platform, we had quite high interclass correlation. Now, we were worried to start with that this might be too high and the data might be too good. So then we had started looking at the association between inflammatory markers, age, various comorbidities, anti-inflammatory medications to make sure that the data was representing and acting as it should, and all the correlations were in the correct direction. So we've been really happy with Quanterix on this in terms of both very few missing data points, excellent ICCs, and data in the expected direction. So with that, I'd like to just thank many of my collaborators, thank Jeff Dage from Eli Lilly for running the plasma tau assays for us, and then also for Simoa with working with us on several of these studies.
Well, thank you, Michelle. So we have time for questions now, and there's been a few questions come in. Before we do so, I figured maybe it's helpful to provide a little bit of feedback. It's kind of music to my ears to hear that you know, Michelle, this being figured that the data may be too good. That's what you said. So maybe to provide a little bit of context here with regard how the assays are performed, if there have been a number of technical questions. So Simoa, what we call single molecular array technology, in short, Simoa is a bead-based assay where typically a capture antibody or capture reagent is coated on beads, and then your detector antibody is biotinylated, and these immunocomplexes, it's a traditional sandwich ELISA, are isolated into small wells in an array, hence the name Simoa, single molecular array. And you'll see, you've seen this on one of the slides, and this will also become clear in some of the publications which will be forwarded to you. And by isolating these immunocomplexes on beads into these small wells, these wells have a volume of 50 femtoliter, and only a single bead with immunocomplex can fit into these wells. Ultrasensitivity is accomplished or achieved. These assays are run on an automated analyzer, so that these are designed with the concept of samples in, results out. Again, it was on one of the slides. And there is a menu of commercially available assays for a number of different therapeutic areas with particular focus on CNS, and that was the focus on today's presentation here as well. And this includes the human tau assay, which was utilized by Michelle, and we have subsequently also commercialized an assay for the murine tau, which was developed by Stephan and colleagues originally through the homebrew program. And that brings up another important point, and we'll come to a number of the questions before. The advantage of the Simoa platform is that this is what we call an open platform as well. So if you as a researcher, customer, have an existing ELISA-based assay, a bead-based assay on the Luminex, as Stephan alluded to, there is an easy transfer of the assay into the Simoa format, into a bead-based format, then with the isolation of the well on a disk, actually, which harbors the arrays here, the single molecule arrays we described as well. And this, as you've seen in the example Stephan has provided, has resulted in an increase of sensitivity from a mesoscale assay, it's a different set of antibodies of 500 picograms per milliliter, to a limit of detection and quantification below a picogram per milliliter, so roughly 500 to 1,000-fold. And I think, obviously I'm a bit biased here. This is a remarkable increase in sensitivity, which really has opened up new frontiers of research. In this particular case, the detection of tau in CSF, murine tau in CSF as well. What I also wanted to say that with regard to volume requirements, and Stephan will discuss this in a second here as well, working with murine CSF, this is very limited from a volume perspective, 5 microliters thereabouts. These assays can be multiplexed either in a commercial format, so off the shelf, have been done for various cytokines and neuronal markers as well, and this can also be done by customer in what we call the custom assay development format, or homebrew. We call this the homebrew format as well. The way these assays are multiplexed, that there's an additional dye encoded onto the bead, so now you have not just a readout of your signal representing your analyte or quantification of your analyte, but you also have representation of the actual analyte, which was captured in this particular case by a capture antibody and then detected by a detection antibody. So with this, what I wanted to do, and please feel free to add questions to the chat room here, to start with a number of questions. The first question was for Stephan, and that was kind of the prelude in the introduction I gave here. Question was, can traditional ELISAs or other platforms be used to screen or select the antibodies for subsequent transfer on Simoa, and what was the rationale to use Luminex to screen the pair of antibodies? I think you talked about this, but maybe it would be good to reiterate what some of the historic perspectives were and some of the experience and lesson learned now as you've worked, you and your colleagues, with the technology.
Yes, sure. The reason to take Luminex is basically, well, it's at least already more sensitive than traditional ELISA. So to go with traditional ELISAs is probably not the way you would do. If you really want to test different combinations and orientations of your antibodies that you have. One reason was definitely also the multiplex capability of Luminex that we had, and this was at the NMI where the the institute in Reutlingen, where our collaborators, which did a major work on this assay development, I have to say, they have already a long-standing experience with this platform, and therefore it was also straightforward, and it would have been much more expensive to do all these stuff on Simoa directly. So that's the rationale behind it, why we took the Luminex platform, basically.
And maybe from your experience in developing a given homebrew assay, now that you have a peer, I mean, what are some of are there any challenges, the ease of transfer of the assay? Maybe if you wouldn't mind speaking about or talking to this as well.
Well, the one experience that we had is that not necessarily then the best antibody pair on a Luminex, or I would expect also on comparable platforms, would also be show the best performance on the Simoa platform, because the antibodies have to be good enough to really be useful in this highly sensitive platform. And therefore we also experienced that we had in the end, we had a different antibody pair than we actually thought from the Luminex data. That was one thing, but otherwise, I mean, it was pretty straightforward. I mean, we were really lucky that we had plenty of good antibodies, and which are also included in other assays, and that we had the opportunity to freely combine them. Mm-hmm. One of the major advantages.
Okay, and then I think maybe some of the attendees online know about this, but the collection of CSF from mice or Cisterna magna. Maybe if you wouldn't mind, the challenges to do so, what volume is typically obtained, and maybe some of the pre-analytical confounders. I mean, it's technically challenging, I guess that's what I'm trying to say, but it would be good to put this in perspective.
Yeah, it is indeed challenging, although we have now a long-standing experience for several years now, and we continuously improve the collection, but also the handling of the CSF. Also in accordance with, for standardization, what is also done in humans, so in the clinics, with the human samples, so to be as accurate as one can be with mouse CSF. When we talk about the volumes, we from a normal mouse, let's say a wild type mouse, but also in our APP transgenic mice, we would collect also depending on the age, something between 20 and 25 microliters, and we routinely aliquot that into 5 microliters in order to be able to measure different biomarkers, but also in case when something goes wrong with the measurement, that you can have the opportunity to repeat it. So that's normally the volume we deal with tau transgenic mice, and so alpha-synuclein transgenic, you happen to have higher volumes, but not like 30 microliters or so, not much higher.
And then the added benefit of the ultra-sensitivity now by Simoa, as you also pointed out, is really now being in a position to be able to detect total tau and also other biomarkers you may be working on from non-transgenic or young transgenic animals, as you said. So in your case, the dilution for tau or for CSF for the analysis of total tau was 1:80, if I remember this correctly.
This is correct, yeah. That was mainly based due to the fact that you have or you had a pretty high dead volume with the automatic system, so we had basically to dilute 1:80. And we were lucky that with our assay, we were still, when you remember the parallelism assessment, we were still on the safe side with this dilution.
Okay, and maybe kind of mechanistically now, going away from the technical details now, I mean, what is the thinking here in terms of pathways, mechanism of action with regard to reduction of what we call total tau? And there was a related question with regard to species we come to as well as a function of base inhibition. I mean, why do we see this? Is this effect of functional neurodegeneration? Is this a direct, indirect pathway? What is the current hypothesis?
Yeah, I mean, the only hypothesis for the moment with the mechanism is that we think it's the beta amyloidosis, so the aggregation and deposition of A beta in the brain that leads to the increase, and if we prevent that in the mouse model, at least we can also prevent the CSF tau increase completely, which is really surprising, I would say, and also is in favor of this hypothesis that it is really highly connected to the deposition of A beta. What it is, and to what extent the cell, I mean, probably it's axons, because the tau is also in the axon mainly, whether it's axonal degeneration or so. I think there's still a lot to do to look into that and come up with a more precise pathomechanism that is behind this CSF tau increase or the prevention of it. But the base inhibition, since there is no direct effect on CSF tau, as we have shown in the short-term experiment, has to do with the amyloid deposition that can be prevented, and therefore the CSF tau increase is related to that.
Yeah. I mean, as you said in one of your slides, and it's been discussed recently, it really has opened up the venue now for a new assessment of targets with regard to base inhibition in clinical studies, either ongoing or to be completed in the future. And that's quite exciting, I would say. I mean, not as potential in what we call primary and secondary endpoint, but as a mechanistic biomarker.
Maybe I can also mention the big advantage to measure the CSF tau and to look at the values of tau in the CSF compared to A beta. It actually doesn't matter when you collect your CSF and look at the levels. Whereas for A beta, as we saw, there are two different mechanisms. The sequestration that basically leads to a decrease of CSF tau, and on the other hand, there's also base inhibitor that also stops A beta production and therefore decreases A beta 42 or A beta in general. And just to say, the CSF tau is basically not linked to this direct effect, and therefore one can directly collect the CSF and quantify CSF tau. Whereas for A beta, you normally would have to allow the system to equilibrate again in order to that A beta 42 really reflects the A beta deposition.
Okay, returning to the clinical domain, the question I think strategically for Michelle and maybe for myself as well, the data you've suggested is that there is a complex functional relationship between tau and functional measures, executive function, cognitive measures, and demographics as well. I think the good news here is that we are in a position to reliably and specifically and precise measure plasma or tau in the periphery, what we call total tau, and also other forms of tau, and that was a related question in a minute. So, and as we know, Alzheimer's disease or neurodegeneration in general are complex diseases. So a question here was, what are the prospects of approval of tau for an in vitro, what we call an in vitro diagnostics by the TBI? I mean, your data and other related data, including data generated by myself, suggest that as a biomarker on its own, and I stress on its own or alone, this is not sufficient as a diagnostic biomarker. Maybe this is not so surprising because we've seen similar data for CSF now as well. So in your view, I mean, is there a role of tau alone without combination with let's say A beta 142 and phosphorylated tau as an in vitro diagnostic, and what are the efforts to have this approved or cleared as an in vitro diagnostic?
Yeah, so I think based on our data and which gels with Heinrich's data and some of the work in ADNI as well. I mean, cross-sectionally, plasma tau alone is not going to be a diagnostic marker for Alzheimer's disease. It will be interesting to look at plasma tau in some of the tauopathies and tau mutations to see if those are sky high, then maybe it could be used in that regard, but not specifically for Alzheimer's disease. I think what we're looking at more is that it's a potentially more of a prognostic risk factor or biomarker, depending on how you define what a biomarker is.
Yep. And then from an approval perspective, one would say that the efforts are currently in the human world focused on CSF just from a historic legacy perspective because of the wealth of data we've generated in CSF. Isn't that a fair statement, or you view this differently?
No, I agree with that.
Question to you both, and we talked briefly about the different assay formats here, is do we know if the species of plasma tau in cardiovascular disease and in neurodegeneration, specific Alzheimer's disease, are the same? Are there differences in the phosphorylation side, so in phospho-tau? I don't know who wants to go first.
I can. I don't think we know that yet. We're just starting to get to that point. It's been interesting with all the tau PET data that have been coming out and the different tau tracers, and I think that's helping us to really understand all the differences in tau processing, and that's different processing may be related to different diseases. Mm-hmm. And so, really, plasma tau is obviously way behind what's going on in the CSF and brain, and so we're going to have to figure that out down the road. I think it is interesting, though, that we are seeing some of these associations with cardiovascular disease as well, which is more, plasma tau may be more of a neurodegenerative, just overall measure, which, even if not specific, is actually a pretty good thing because we don't have anything right now to indicate who might progress and who might not.
Okay. And then, Stephan, I think one of your slides, you showed that there are multiple bands for what we would consider total tau now detected by these various antibodies, and you mentioned N-terminal truncation. So in the murine world and also maybe in the human world, what do we know about truncations, different species in CSF or blood of tau?
Well, what we know at the moment, that's for CSF. What we know is that the main species that are present in the CSF of humans, because that's actually the only studies I'm aware of, is really N-terminal fragments, and there is no full length, as it looks like, or C-terminal fragments. At least assays that would capture those species do not show anything. There has been more recent data with mass spec where one could assume that there might also be a tiny fraction of C-terminal, but the predominant one is really the predominant species are really the ones that are in this proline-rich domain. However, in blood, as far as I know, one has no clue what is there, and that makes it also difficult to really be sure that when we use such an assay, do we really measure the same thing? Because assuming that there might also be full length around whatever reason in the blood or so, but there might be some peripheral source, whatever we are not aware of, that also has different species. That also can mask your effect from the CSF tau that might still be present in the blood, but just does not come up because there is a lot other tau that is coming from the periphery. So therefore, I think now we have, to a certain extent, we have solved the sensitivity, so to say, but in the future we have to focus more again on selectivity in addition. So that's the biggest challenge, I think.
And what do we know about phosphorylation side with regard to p-tau? And again, this comes in different forms of phosphorylation, 181 being the most traditionally or most frequently studied, 231, and then some more C-terminal phosphorylations. Are there any data in mice with regard to abundance of these phosphorylation sites? And would we expect differences now as, say, either as a function of aging or as a function of pharmacological manipulation of base activity?
In the CSF or blood, there's no data on it due to the lack of assays, actually.
Yep.
We are working on that, I guess, as others do as well. And of course, we are now talking about the APP transgenic mice. It's something different. In tau transgenic, there you might find
yep.
it.
Okay.
But still, you have to have the assays.
Yep. This brings up a question here. So the Simoa tau assay now measures what we call total tau, not phosphorylated tau, forms of tau. But there is an assay which will be made commercially available for what we call phosphorylated form of 231 at residue 231 and 181 of total tau as well. So these are separate assays, but these will be available as Simoa assays as well. So maybe then kind of to finish up on this, there was a final question. What is the verdict about other modality now other than detecting of brain pathology-related proteins such as tau? What is the role of microRNAs? They've also been studied in AD. I mean, do we have comparative data if they have a diagnostic role in AD, or could be used as a potential readout of target, this being base inhibition in mice? Well, these data are still to be generated.
Michelle, would you go? Or I mean, I just think, of course, it's always promising to come up with other biomarkers, but in the end, at the moment, there's still the A-beta-42 and tau and phosphorylated forms of tau that are the best investigated and best established biomarkers for AD.
Michelle, is there anything you wanted to add, or?
No, I would agree with that. I mean, I think there's a lot of other platforms that we need to explore, but building off, again, the CSF, A-beta, and tau, and with the work Stephan's doing and trying to see, what we can find out with plasma, I think is important as well.
Okay. Well, maybe a comment to add to this, and you'll see this in an upcoming publication, that Simoa has also been used to detect microRNAs as well, so it's not just proteins, but it can also detect without amplification ultra-sensitive way microRNAs or other species of RNA. So with this, I want to thank the speakers again, Elizabeth as the technical moderator as well, and all the attendees. The slides will be made available. Some of the data which have been presented today have been published as well, so please feel free to reach out to Quanterix for a follow-up of either with regard to the publications or any other related questions. And with this, thank you again. Have a good weekend, and we'll talk to you soon.